Alright, so this video is just going to explain some of the labeling here and a little bit about the definitions of each one. So we're looking at a plant cell here. So some of the characteristics, how we know it's a plant cell, is the cell wall here and the chloroplast here. Those are and the large vacuole here. Those are kind of three characteristics of plant cells. So the cell wall it's not so much for protection, but it gives a rigid layer that allows the cell to absorb a lot more water than it normally would, because that pressure that water exerts on those walls is what allows a plant to kind of stay up, basically. Chloroplast is where photosynthesis will take place. The vacuole is a storage thing here, so it stores a lot of water. It can also store starch and things and glucose from photosynthesis. So the rest of these are pretty typical for plant and animal cells. So cytoplasm is kind of the liquid part of the cell. The nucleolus is kind of the part of the cell that this is all basically DNA. So when you look through a microscope, sometimes you see a dark spot kind of in the nucleus. That's the nucleolus. And that's where the DNA is condensed at. So a couple other ones here. So the smooth ER here is where lipids synthesized and a few other things like that. While over here with the rough ER, and you see these little dots here, that's kind of the definition of the rough ER. And that is where ribosomes are attached to. Okay, and that's what these are pointing here. Now ribosomes can also be along here, and we call those free ribosomes. So if they're attached to the rough ER, they're called they're attached ribosomes. If they're free from the rough ER, they're for free ribosomes. Now the rough ER produces proteins that then those proteins are going to be transported out of the cell, while free ribosomes generally make proteins for use inside the cell. Okay, so here's the nucleus, right? And you kind of think about that as like the control center of the cell. This is where the DNA is at. This is where that DNA is changed into RNA, which then is used to make proteins. On the mitochondria, you kind of think about it as the powerhouse of the cell. This is where ATP production takes place. And its kind of distinguishing factor under a microscope is this squiggly line. So keep a lookout for those squiggly lines. A lysosome, which we'll talk about later, is kind of a bag of enzymes, and this kind of digests things. The Golgi body here is used as part of the transport, so generally proteins made in the rough ER are transported to the Golgi body and then out of the cell. The Golgi body and the rough ER, or the smooth ER, look very similar, so just kind of be aware of that. The chloroplast, we talked about those, but their kind of distinguishing characteristic is they look like, you can't see it so well in this picture, but it looks like little stacks of pancakes. You can see those kind of over here. That's kind of how you tell a chloroplast from a mitochondria. Okay, so that's the plant cell. Now an animal cell looks very similar, but there's a couple of additional things which I'm going to talk about. I'm not going to repeat all of these unless I remember something I forgot. So one is kind of, some can have cilia. So look at, these are just little protrusions of the cell membrane. So these can be used, like for example, in the lining of your lungs. It's a cilia to move mucus up out of the lungs, and then you kind of cough it up. They can be used to kind of move things along. Microvilli here do a couple of different things, but one of the things they do is they kind of increase the surface area. So if you have microvilli, you can increase the amount of material you take in, and we'll talk about that later. The rest of these are pretty much the same. Notice that a plant cell doesn't have a cell wall. It only has a cell membrane. Okay, so that's a distinguishing characteristic. The other thing is plants or animal cells have centrioles, and these are basically used in mitosis. Okay, so you might have gotten that from Bio 2 if you remember, and they're only found in animal cells for the centrioles. Okay, the rest of these, Golgi apparatus, smooth ER, mitochondria, so forth, are pretty much all the same. Our last thing here is bacteria, or prokaryotic cells. Okay, these are some different shapes of them. You don't need to worry about that. You do know that they reproduce by binary fission. That's one of their characteristics. Generally speaking, prokaryotic cells are simpler, but still incredibly complex. We're still learning a lot about them. So they have some things. So they have a cell wall, but this is different from a plant cell wall. It's made of something called peptoglycin. I'm probably spelling that wrong. But, while a plant cell wall, plant is made out of cellulose. So that's kind of the difference there, and you need to check the name of that. I've forgotten at the moment. But plants are made out of cellulose. So that's one difference. I notice they have cytoplasm, ribosomes. They have what we call a bacterial chromosome. And there's something called a plasmid. So bacterial chromosome is just one big piece of DNA, one big loop of DNA, while plant and animals have, if you remember, chromosomes. Right? So that's kind of a difference is here. Plasmids are tiny pieces of DNA that actually bacteria can trade with each other, and these are what kind of can lead to bacterial resistance. We'll talk about these in a lot more detail when we talk about biotechnology. The flagellum are used to move. Not all bacteria will have a flagellum, but some do. Some animal cells will have a flagellum also, just be aware, but the shape of the flagellum is unique for bacteria. And finally, the capsule, which are these kind of little squiggly things here. Those capsules are involved in the idea of this is how things how the bacteria attach to things, okay? So in some cases, it's these capsules that allow bacteria to infect you. If you get a bacterial infection, these capsules, for example, connect to cells in your sinus, can cause a sinus infection, that sort of thing, okay? So that kind of gives you an overview of the shapes and things. So you should be able to draw each one of these, okay? It doesn't have to be perfect. It's not an art class, but you need to be able to draw these and name most of the organelles. All right, thank you.